Good evening guys, and today we are going to discuss about the acute inflammation. It's the second part of the second chapter of the pathoma, which is the inflammation, inflammatory disorders and the wound healing. So we have discussed about the first phase of acute inflammation, that is the fluid or edema phase, in which the fluid starts to accumulate in the interstitial tissue, and the second phase of the acute inflammation is the accumulation of the neutrophils. Okay, and it occurs around about 24 hours after the start of the inflammation, and the third is the macrophages, which occurs after 2 to 3 days. So let's discuss about the neutrophil arrival and function. And in general, it can be the leukocytes, because leukocytes can also be include macrophages, that are monocytes. So we are talking in general about both leukocytes and in specific about the neutrophils. Arrival and function has several steps. Now we are discussing about 5 major steps, and in the next video we'll discuss about the remaining steps. First is the vasodilation. In the previous video, I have told you that vasodilation occur at the level of the arteriole, and increased vascular permeability occur at the level of the post capillary venule. It means that when the vasodilation occurs, the blood starts to move slower in the post capillary venule, and in normal conditions, the cells move in the center of a blood vessel. But when there is vasodilation, when the blood starts to move slowly, the cells also starts to marginate from the center towards the periphery. It means that, for example, this is the blood vessel. Okay, and the cell starts to move towards the periphery of that blood vessel. They starts to move from the center towards the periphery. The second step is the rolling. What is rolling? Here, two important things you should keep in mind, that there are two types of like molecules that are upregulated on endothelial cells and the leukocytes. What are the molecules that are expressed on the endothelial cells? Or these are known as selectins. And what are the molecules that are upregulated on leukocytes? It's the sialyl Lewis X. Okay, it's sialyl Lewis X. I will explain you that. First of all, the selectins act as the speed bumps. Okay, speed bumps, for example, in the roads we have seen the speed bumps that are upregulated on the footpaths, the pedestrian pathway, so that the car starts to move slower there. So as same scene is occurring here, that selectins are speed bumps that are upregulated on the endothelial cells, and they have role in the rolling of these leukocytes. There are two types of selectins. First is the p-selectin and the second is the e-selectin. P-selectin is produced by the Weibel palade bodies, and Weibel palade bodies are present. These are some sort of granules that are present in the endothelial cells. Okay, and this starts to secrete two substances. First, we have discussed p-selectin, and the second important substance secreted from the Weibel palade bodies is the von Willebrand factor, and von Willebrand factor have some role in the clotting of the blood. We'll discuss them later. So up to this you have learned that p-selectin is secreted from the Weibel palade bodies, and it is upregulated on the endothelial cells. And this secretion, or this all of the process, is mediated by which mediator? It's histamine. You have to remember that. Okay, p-selectin is mediated by the histamine. The second type of selectin is the e-selectin. E-selectin is also produced and is upregulated on the endothelial cells, and it is induced by the tumor necrosis factor and the interleukin 1. We have discussed in the previous video that TNF, which is tumor necrotic factor, and interleukin 1 has very important role in acute inflammation. when discussing about these selectins have been upregulated, right? They have been upregulated on the endothelial cells. But to which molecules these selectins are going to bind? They are going to bind on the Lewis X on the leukocytes. Lewis X on the leukocytes. And interaction between the selectin and the Lewis X will result in the rolling of the leukocyte along the vessel wall. Are you understanding that? We have came across this step. It's rolling, the upregulation of selectins on the endothelium and binding it with the Lewis X on the leukocytes result in the rolling of that particular leukocyte. Third, function. The third step in the arrival and function of neutrophils or in general leukocytes is the adhesion. So there are two types of molecules that will be upregulated on first endothelial cells and the second the leukocytes. What substances are upregulated on the endothelial cells? These are the cellular adhesion molecules, which is CAM. Cellular adhesion molecules will be upregulated on the endothelium. Okay, it will be upregulated on the endothelium. And what will be upregulated on the leukocytes? These are integrins. Integrins will be upregulated on the leukocytes. What are the examples of cellular adhesion molecules? It can be ISAM and VSAM or VCAM. You can pronounce it either ways. ICAM and VCAM are upregulated on endothelium, and the integrins are upregulated on the leukocytes. Okay, and the regulation of the SAM molecules is by the tumor necrosis factor, interleukin 1. We have discussed that these both mediators have important role in the acute inflammation, and integrin upregulation is by the mediators, the complement protein 5A and leukotriene B4. You have to remember these mediators as far as the acute inflammation is concerned. So the interaction between the SAM or CAM and the integrin result in the adhesion of the leukocyte to the vessel wall. Okay, we are concerned with adhesion, right? So we have discussed up to here. Arrest and the adhesion. Adhesion occurs due to the interaction between the cellular adhesion molecules on the endothelium and integrins on the leukocyte. So they result in the firm adhesion of the leukocyte to the vessel wall. Talking about the adhesion, we can discuss a disease which is known as the leukocyte adhesion deficiency, which is LAND, and it's not very common among the population, but it's due to the autosomal recessive trait, and it results in the defect of integrins. Integrins are upregulated on leukocytes, right? This is the integrin that has been upregulated on the leukocyte. But in case of leukocyte adhesion deficiency, there is defect of the integrins, particularly which part of the integrin or which integrin. It's CD18 subunit of the integrin that has been defected in this disease. So the clinical features of this disease are will be according to our concept. So what does it say? It says that first clinical feature is the delayed separation of the umbilical cord from the fetus. What does it mean? It means that when the leukocyte adhesion deficiency is there, it impairs the ability of neutrophil to adhere to the blood vessel, right? Because integrins are not there. So how the neutrophils gonna adhere to the blood vessel wall, and they cannot migrate to the site of infection. It results in the delayed healing and separation of the umbilical cord. Are you understanding? Because neutrophils cannot adhere to the vessel wall, they cannot migrate to the site of infection. It results in the delayed healing and the separation of the umbilical cord. Second feature, clinical feature of LAD is the increased circulating neutrophils. You need to remember that there are two types of neutrophils. First are the circulating neutrophils, okay, and the second is the marginated pool of the neutrophils. What are marginated pool? 50% of the leukocytes are present in the blood vessel walls, in the lungs, in spleen and liver. They are just there that whenever they are needed, that pool is already ready to be delivered. Okay, so rather than freely circulating in the bloodstream, they are attached to linings of the blood vessels, lungs and liver and spleen. So, what does the leukocyte adhesion deficiency result? It results in increased circulating neutrophils, because as I have told you that these leukocytes cannot adhere to the vessel wall, they cannot pass through the vessel wall. So there will be neutrophilia. It means that a lot of neutrophils will be accumulate in the bloodstream. The third is the recurrent bacterial infection that lack the pus formation. What is pus? Pus is basically the dead bodies of the leukocytes. I cannot simplify that enough, that pus is the dead bodies of the leukocytes. When the white blood cells or leukocytes or neutrophils cannot emigrate, cannot pass the blood vessel, cannot adhere to the vessel wall, how they can come into the tissue space? They cannot. So there will be recurrent infections, but they will lack pus because there are no neutrophils there, because the neutrophils are lacking the integrins. The fourth step of this process is the transmigration and the chemotaxis, in which the leukocytes transmigrate across the endothelium of the post-capillary venule. We are discussing this all process in which location? It's the post-capillary venule, and these leukocytes will start to move towards the chemical attractants, i.e. the chemotaxins. Okay, and this process is called as chemotaxis. These leukocytes, neutrophils, either the macrophages, any of these leukocytes move towards the chemical attractants, which are either produced by the bacteria or are present in that tissue. So this movement is known as the chemotaxis. The four important chemotaxin molecules that cause the neutrophil movement, that cause the neutrophil chemotaxis are, I have been mentioned them in the previous video, so let's revise them. First is the bacterial products. Okay, then second is the interleukin 8, the complement C5a and the leukotriene B4. IL-8, C5a, leukotriene B4 and bacterial products cause the chemotaxis of the neutrophils towards the tissue. The fifth step is the phagocytosis. Phagocytosis, we are here, right? We are here. We have transmigrated across the vessel wall and we are moving towards the chemoattractants in the tissue that are already present in the tissue. So now the leukocyte will cause the phagocytosis of this debris or bacteria or necrotic tissue, anything. Consumption of the pathogens or the necrotic tissue, it's the phagocytosis, and it's occur via the opsonization. What is opsonization? Opsonins are special kind of proteins or tag proteins, for example IgG or C3b. These both are important examples of the opsonins. Opsonins are like tag. For example, I have tagged this molecule with IgG, so the leukocytes see the IgG molecule and they recognize that this molecule is to be phagocytosed. For example, this substance or debris is opsonized by C3b. It means that the leukocyte identify the C3b and recognize that this molecule should be phagocytosed. So this is the tagging process of the debris or opsonization. Pseudopods are extended from the leukocytes. It means that, for example, this is a leukocyte and pseudopods means that their arms will be emerged from the body and they will include the bacterial product or they will include the necrotic debris, and this forms the phagosome. It's the phagosome and it will combine with the lysosome and it will form the phagolysosome, and in the lysosome we have the hydrolytic enzymes and that will cause the digestion or phagocytosis or the hydrolytic process of that particular debris which have been included in the phagosome. So talking about this phagolysosome, there is a very special disease or syndrome, I must say, which is known as the Chediek Higashi syndrome. It's the protein trafficking defect and it's also autosomal recessive. We have discussed about leukocyte adhesion deficiency, LAD, which was also autosomal recessive, and the Chediek Higashi syndrome is also autosomal recessive, and in which there cannot be the formation of phagolysosome. There is impaired phagolysosome formation. So let's discuss about its clinical feature of this Chediek Higashi syndrome. First is the increased risk of pyogenic infections. It means that there is defect in the phagocytosis, the lysosomes in the neutrophils. Okay, the neutrophils have lysosomes. They are unable to fuse properly with the phagosome. Okay, the phagosome contains the bacteria. Okay, the phagosome contains bacteria or they contain the necrotic debris, but they are unable to fuse properly with the neutrophilic lysosome. So the resulting ineffective killing of the bacteria. So there will be infections with pus. Okay, there will be increased risk of pyogenic infections. Second is the neutropenia. Neutropenia occurs because the formation of abnormal lysosomes. There will be formation of abnormal large rhizosome inside the neutrophils. These are known as the giant granules, and these granules impairs the neutrophil function and survival in the intramedullary tissue. It means that when neutrophils are being formed in the bone marrow, they are premature. They die prematurely, okay, due to the formation of the giant granules inside the neutrophils. The third clinical feature of Chediek Higashi syndrome is the giant granules in leukocyte. As I have discussed, that giant granules starts to form in the leukocyte and they fuse. There are a lot of granules that are fused together to form the large or giant granule, okay, and they impairs the normal function and the survival of that particular leukocyte. The fourth is the defect primary hemostasis. The hemostasis or the blood clotting is the function of the platelets, right? So platelet is also a blood cell. It's also a blood cell. Inside the platelets, there will be formation of dense granules and they will impair the function of the platelets, that is hemostasis. So there will be defect in the primary hemostasis. Fifth, the clinical feature is the albinism. Albinism is like a whitish or grayish appearance of the hair of head. It can be hair of eyebrows, eyelashes, and it is due to the melanin is trapped within the melanocytes in like granules known as melanosomes, and these melanosomes prevent the proper distribution of melanin to other cells. Basically, Chediek Higashi syndrome is a protein trafficking defect. It means that the migration or the trafficking is blocked inside the cell, so they cannot transport the materials, they cannot receive the materials properly, so melanin gets trapped inside the melanosomes, inside the melanocyte, and these melanocytes cannot transfer that particular melanin to other tissues, other cells, so there will be clinical symptoms of albinism as well. The last is the peripheral neuropathy, and in this condition, our peripheral nerve starts to bring the signals towards the central nervous system, right? But the signal is transported via the Schwann cells inside the nerves, inside the axon of that neuron. So there will be accumulation of the giant granules in the Schwann cells. Basically, in all of the clinical feature, there are some sort of granular involvement, right? In case of increased risk of pyogenic infection, there will be large lysosomes. Neutropenia, also giant granules. In the defect hemostasis, there will be also granules in the platelets. And in albinism, there will be melanin trapped. And in peripheral neuropathy, giant granules in the Schwann cells so that the nerve signals cannot be transmitted well. So we have discussed 5 steps of the neutrophil arrival and function: the vasodilation, rolling, then adhesion, transmigration, chemotaxis, and the last is the phagocytosis. And in the next video, we'll discuss the remaining steps of that acute inflammation and neutrophil arrival and function. And I will see you in the next video. Till then, goodbye.